All right. Lovely. Looks like things are slowing down. So I will officially welcome you all. Good mid-afternoon, as I've always said, and welcome to the Mid-Afternoon Masterclass, the fifth one for the week, as part of the University of Melbourne Science Festival and National Science Week. My name is Julia, and I will be your host today. Before we begin, I do want to acknowledge country. I want to acknowledge the Wurundjeri people of the Kulin Nation as the traditional custodians of the lands on which I am on today, and also acknowledge the traditional custodians of the lands on which you, our audience, are all zooming in from as well. I want to pay my respects to elders past, present and emerging, and extend that respect to all Indigenous people in the audience. And really importantly, at this time of National Science Week, I also really want to acknowledge and respect that Indigenous, Aboriginal and Torres Strait Islander people have been researching and learning and caring for this beautiful land for tens of thousands of years. And so they are our first scientists. So today, our fifth mid-afternoon masterclass. Welcome back if you have been here before. If this is your first one, welcome. These events have been happening all week, and it's a quick little 45-minute, little bite-sized element of science. We invite an academic to talk on some fascinating topics. Each session will be, and this one will be for about 45 minutes. It will include a short presentation, and then we'll be going over to some questions as well. So put your thinking caps on and think about any questions that you might want to be asking our presenter all the way through, and I'll explain how you can do that soon. So today, the event, we're all in. I'm very excited about it. It's all going to be about vaccines and vaccine development then and now. We're going to find out about the history and the science behind this life-saving research. To speak on this topic, got pleasure in welcoming Dr. Jennifer Juno from the Doherty Institute. Hi, Jennifer. How are you going?
Good, thanks. I'm really excited to be here.
That's good. I'm excited for you to be here too. So to let everyone know about Jennifer's background, so she obtained her PhD from the University of Manitoba. If those had a clear eye, a clear ear there is a Canadian accent that you might have heard. So she studied, in University of Manitoba and looked at the immune dysfunction in the chronic HIV infection. She's done some postdoctoral work at the Public Health Agency of Canada. And then in 2016, had the call of Australia and joined Dr. Stephen Kent's lab at the Doherty Institute. And so she's been continuing to study the immune response of the HIV infection, and also now has an interest in the flu infection and vaccinations in general. So that's your overall bio, Jen. But please, I'd love to, before I sort of go into a few housekeeping things, just love to hear from you what it is about this topic that fascinates you so much. I mean, I think we've had a lot of registrations, so clearly it's a fascinating topic to a lot of people right now, but what is it for you that made you decide to follow this path?
Yeah, as you mentioned, I started doing my PhD in HIV immunology, which obviously is an area where we've been looking at vaccine development and trying to understand how to design better vaccines for decades now, effectively. And that was really fascinating and rewarding research, but you always feel like you're sort of a much smaller piece of a really big puzzle, and that there's a community of science sort of building towards better understandings of things. And in the last few years, having moved more into influenza work and more recently COVID-19, it's just been this absolute shift in seeing how immediately research can have an impact on the real world around us. And that's just been an absolutely phenomenal experience. So it's been a really interesting journey. And my hope is that we can take lessons and learn from all of these things in this fantastic pace of vaccine development, and now go back and apply these things to some of the challenges that we've been facing for many, many years. I think it's a really exciting time in science, notwithstanding a bit of the chaos that's been happening globally the last year and a half or so.
Absolutely. I think an exciting time is an understatement. I was saying that being part of the Doherty Institute over the last 18 months just must have been an absolute wild ride. But thank you for all your work so far, and I can't wait to hear more about it. So before I throw it over to you, I just want to let everyone out there know. So as I said, there's going to be a Q&A session at the end of the presentation. So please feel free to put in question. So to do that, there is a Q&A button at the bottom. So if you sort of toggle your mouse a little bit, there's a bar. If you haven't been on Zoom before, there's a bar at the bottom. If you just click on Q&A, just pop your question in there. I do also want to mention, obviously, this, as many health topics are, it's quite an emotional topic. So please really respect the topic and what we're talking about right now before deciding on what question or questions that you'd like to ask. I think that's it. That's all from me. I'll get off your screen now. But thank you so much for joining us, Jennifer. I can't wait to hear all about it. And I'll throw over to you now.
Thank you very much. All right, are we sharing properly? And we're
yes, all good.
Fantastic. So thank you so much for everyone for signing up and coming today. When we were first talking about topics that we could talk about in science and immunology, I sort of raised the idea of whether we wanted to talk about vaccine and vaccine development and whether people would just be completely over it and completely tired of hearing about these things. So it's really encouraging, and I'm really glad to hear that there's been a lot of interest in this topic. So today, I really want to talk about not just the vaccines that we're all very familiar with now related to SARS-CoV-2 and COVID-19, but really try to give an overview of where we've come from in this field and how we were able to accomplish what we've accomplished within the last year and all of the lessons that have built up to being able to respond to the current pandemic. So I want to talk a little bit about the history of vaccination, how we start with a story about cows and end up in stories of very, very sophisticated and technologically advanced vaccines. And then, of course, we do want to deal with tackling SARS-CoV-2 and how did we go from a very, very brand new virus that we didn't know anything about to very rapidly being able to design and produce quite effective vaccines that we've been able to roll out across the world within such a short period of time. And then using this information, I think we can ask a lot of interesting questions about where we're going to be able to go from here. What are the pathogens that we might encounter in the future? How are we going to handle the likelihood of additional pandemics or pandemic threats? And how can we try to develop vaccines that give us the broadest protection and the longest lasting protection possible? So some people might be familiar with the story. It's a story that we typically teach in school and teach students about the history of vaccination. And we often talk about Edward Jenner in 1796 and how this was the first person who really started the field of vaccination. And the story revolves around smallpox, which was a major public health threat at the time, and the sort of widespread knowledge that milkmaids who came into contact with cows would often get an infection with cowpox, but wouldn't seem to become infected with smallpox. And so this led to the idea that perhaps the cowpox infection was protective and would give your immune system knowledge that would stop you from becoming infected with smallpox. So Edward Jenner went ahead and actually collected some pus, essentially, from a wound from a milkmaid that was infected with cowpox and inoculated or scratched that material into the arm of a young boy. And I will say we have come a long way since 1796 in terms of the idea of human clinical trials and informed consent. But in this case, this was what happened. He inoculated the young boy with cowpox. The boy did develop an infection. But then Edward Jenner came back and later tried to infect the boy with smallpox, and the boy was protected from infection. And this is to be considered, one of the first examples of vaccination leading to protection from an infection. But of course, as is often the case with stories that we like to say are the first example of something, that's actually not necessarily the truth. So if we go and we look further back in history, there were actually many other examples of other people and other groups that were performing essentially vaccination long before this. So there are accounts of smallpox inoculation across China and India from as early as the year 200 right up through the 1600s. And in these cases, they weren't using cowpox, they were actually using a small amount of smallpox, again, to inoculate individuals, often children, to give them a very mild infection so that they would be protected from more severe infection later in life. And this was actually quite a widespread practice throughout China and India. And then later on in the early 1700s, again, some of the aristocracy came across this practice in Turkey, and again had their children variolated or vaccinated to protect them from smallpox infection. So over and over again, we see this idea of educating your immune system about a pathogen to protect you from infection later in life. So no matter where we want to ascribe the early starts of vaccination, although it was clearly much earlier than we normally think of it, ultimately this led to the global campaign from the WHO to eradicate smallpox. And smallpox was declared eradicated in 1980 following this widespread vaccine campaign. And so the main takeaway message here about the idea of what we think of as vaccination is that a pathogen similar to a major global public health problem like smallpox was taken from the cows, this vaccinia virus. And if you expose your immune system to it, it teaches your immune system enough of a lesson that it knows how to respond and defend you from a later infection. And after that vaccination, you're then protected against infection from the dangerous pathogen, which in this case was smallpox. And this is really the goal of vaccines, no matter what pathogen, what infection, anything that we're talking about. We want to educate your immune system in a safe way so that it can respond to a challenge from that pathogen later. And I don't want to get too much into heavy jargon or immunology, but I do just want to recap really briefly how our immune system responds to pathogens. So in this case, if we think about a virus, especially like influenza or SARS-CoV-2, we're exposed to the virus potentially in our nose, potentially in our bloodstream. And no matter what, we have immune cells whose job it is to collect those viruses. They have sticky little arms that wave around and they can pick up pieces of this virus. And it's their job to take them and travel them through our body to our lymph nodes. And in the lymph node, these cells will present the virus to our white blood cells from our immune system. And they'll say, hey, check this out. And this is something not supposed to be here. We need to mount a response to this. And then two of my favorite white blood cell types called B cells and T cells will recognize those little pieces of the pathogen and say, okay, we need to start reacting to this and we need to start making proteins and responding. So these cells will then make a number of different molecules. And the one that we're going to talk a lot about today is antibodies. And I'm sure we've all heard a lot of things again about antibodies to SARS-CoV-2. And antibodies are small pieces of protein whose job it is to attach to a virus. So they can stick on the outside of the virus. And by doing that, they'll actually prevent the virus from being able to come and infect our cells. So after the immune system has been activated, it's learned things about this pathogen, then these cells can go back out into our bloodstream and go out into our tissues and protect us from future infections. So where did we go in terms of vaccination after the development of the smallpox vaccine? Well, the first series of vaccines, essentially, that were developed, were all falling into this category of what we call a live attenuated vaccine. So this means the vaccine is based on a live pathogen. It's able to replicate, but it's been weakened in some manner. So in this case, that's like using the cowpox virus instead of the smallpox virus. And we use a similar approach for early versions of vaccines against rabies. We still use it actually for the yellow fever vaccine, which is one of the most effective vaccines that we have available. It's used for the BCG vaccine, and again, early versions of the measles and mumps and rubella vaccines. And this worked for a number of these pathogens. Obviously, it stimulates a good immune response because there is a pathogen that's replicating, even though it's not causing disease. And that's a really good way of activating your immune system. However, sometimes the side effects from those vaccines can be a little bit stronger than what we would like. So people moved on to different versions and different vaccine platforms as technology started to evolve. So the next class of vaccines that gained popularity were vaccines that were killed or inactivated. And this includes vaccines initially developed for typhoid, cholera, pertussis, and several encephalitis viruses. And in this case, there's no replication. So the pathogen isn't able to replicate in your body. It's either been killed, so through exposing it to heat or to other agents that kill it, or it's inactivated, so that essentially it can't cause any sort of infection. And obviously, this is desirable for some pathogens, especially ones that might be a little bit too dangerous to use a live attenuated form for. And then things got even a little bit more sophisticated. So we started being able to make purified antigens. And an antigen is just a piece of the pathogen that we want our immune system to recognize. So just think of it as a little tiny part of the pathogen. And this works for other vaccines that we commonly give, so diphtheria, tetanus, anthrax, pneumococcus, and meningococcus. So in this case, we're not even showing your immune system the entire pathogen. We're just showing it a very specific part and saying, hey, this is the part that we want you to pay attention to. This is the part that we really want to generate an immune response. And this tends to be an even more specific approach to give a really strong immune response against exactly the part of the virus or the bacteria that we want the immune system to focus on. Now, in the last few decades, technology has really led to massive changes within the scientific field and also vaccine development. So we've gone from these initial early concepts of live attenuated vaccines or inactivated vaccines. Now we have a whole range of options available in our toolkits. So we can make very, very, very small specific pieces of just tiny, tiny, tiny parts of a virus or a bacteria. And that might be something like a peptide vaccine. So it's just this very specific part that we want to show your immune system. We have something called virus-like particles, and this actually forms the basis of the HPV vaccine, which I'll talk about in a minute. And then we have some of our more modern vaccines. Everyone, of course, now is extremely familiar with the idea of RNA vaccines, and also familiar with the idea of viral vectors. So the viral vector means that we use a very harmless virus to deliver genetic material encoding something else. In the case of COVID, that might be the SARS-CoV-2 proteins, to tell your immune system to make a response against that. And there are several other types of vaccines that I'll talk about, especially in the context of flu vaccine. So we've really seen this enormous array of options that we have now, where for different pathogens or different types of an immune response, we can select exactly what kind of vaccine we want to use. And I mentioned HPV, and I think this is an interesting story to highlight. So HPV, or human papillomavirus, is a vaccine with a nice Australian connection. So Ian Fraser, the University of Queensland, was really instrumental in developing the technology for the HPV vaccine. And HPV was a virus that was really difficult to grow in the lab. So unlike other viruses like influenza or even HIV, it was quite difficult to grow large amounts of the virus in the laboratory setting. And so a technology was developed, again, to just express very specific parts of the virus that we wanted our immune system to generate antibodies to. And when we express that specific part, scientists were able to create these virus-like particles, which is the picture shown here on the left. And this is 72 copies of this one portion of the virus. And it forms this ball and sort of looks like a virus. That's why we call it a virus-like particle. And this turns out to be exceptionally good at activating your immune system. So when your immune system sees these 72 copies of the same thing over and over again, it's a really good signal for your immune cells to say, hey, this is not something that's supposed to be in my body. We definitely need to raise a response to this. And as a result, the HPV vaccine causes an antibody response that is substantially stronger than the response that we typically see if you became infected with HPV naturally. And the immune response that it elicits lasts for many, many, many years, and we think now probably even decades. So this has been a really good specific example of where we designed one type of vaccine towards HPV, these VLPs. It works particularly well. It gives you really long-lasting immunity. And we've now increased these HPV vaccines to incorporate a number of different strains that are all causing cancer. And so this has been a great success story of a very focused vaccine development pathway that led to a really, really strong vaccine that will hopefully be used for many, many years to come. Now, kind of at the other end of the spectrum, we have influenza vaccines. And unlike HPV, where we really focus heavily on one type of vaccine, there are a number of different influenza vaccines that are in development and testing and that are constantly being updated. And I think we're all familiar, of course, with the idea that the flu vaccine needs to be updated annually. This is because the virus is really good at changing the portion of itself that the immune system recognizes. And so we constantly need to be re-educating the immune system to try to keep up with these changes. And there are also some other difficulties that we want to overcome with better flu vaccines. First of all, the flu vaccine is also notorious for having relatively low immune responses in older individuals. And of course, these are the groups of people that we really want to protect the most with a vaccine. So we're looking to try to find ways that we can improve the immune response in people whose immune systems might not be quite as responsiveness. Additionally, we know that because people constantly receive flu vaccines every year, some people end up with high levels of antibodies. And normally this would be a good thing, but it means that sometimes when you receive the vaccine, actually, your immune system kind of ignores it a little bit, and it doesn't actually boost your antibody levels as much as we would like. And so these challenges are something that we're trying to overcome by producing different types of vaccines. So I don't believe it's been tested here in Australia, but there is actually a live attenuated version of the influenza virus vaccine, which can be taken as a nasal spray. And there's been a lot of testing of this in North America and in other countries so that we can try to elicit an immune response to flu that really lives in your nose and your respiratory tract to try to protect you right from the site of infection. And additionally, there's been a lot of innovation going on with the types of portions of the flu virus that we show to your immune system. So this schematic here, shown in the red and the green, is the portion of the flu virus that your immune system looks at and needs to respond to. And the part in red is the part that your immune system reacts to most strongly. So it produces most of its antibodies to try to respond to this red part. However, that's the part, as I said, that the virus can change most easily. So it's constantly switching that around to evade our immune systems. But there's also this part here in green, and the green part actually doesn't change very much at all. It's very, very stable across different strains of flu. And so people are now testing vaccines where we actually just chop the head off of this protein and just show the immune system the green part and just say, hey, only focus on this, only pay attention to this, and produce as many antibodies as you possibly can just towards this green part. And this is an attempt to produce what we would call an universal influenza vaccine so that we don't need to constantly be updating the virus every year. And so clinical trials of these approaches are currently underway. And even earlier this year, there were reports coming out of the NIH of looking at this approach of changing how we tell the immune system about the virus to see whether that can create a more effective vaccine. So with all of this in mind and all of the research that goes into all of these different vaccine platforms, what do we do when a brand new virus comes along? And luckily, we had a very good example of this within the last year or year and a half when we started to see COVID-19. So if you want to design a vaccine from scratch, where do you start? What do you even look at? This schematic shows you a number of the different proteins within the SARS-CoV-2 virus. So there's the spike, and that's the protein that we talk about quite a bit on the outside of the virus, but there are all of these other components as well. So what would we pick? How would we make a vaccine? What would we start with? And what I think was really incredible was actually looking at the timeline of early vaccine development. So on the 10th of January, 2020, the sequences encoding SARS-CoV-2 were released from China. So that was the first knowledge of how we could start to express and create these proteins in the lab and start to study the virus and start to make vaccines. It was 24 hours later that the NIH began to produce stable versions of the spike protein. Now, I'll come back to what that means in a minute, but basically within 24 hours, labs at the NIH were starting to produce in the laboratory this component of the spike protein from the virus. It was only 4 days after that that Moderna actually started to produce clinical grade material for their mRNA vaccine. So they were able to use those sequences, and we'll talk about some of the modifications that they made, and begin to produce a vaccine within 5 days of the sequences being released. And it was just a couple of months later in March where they started their first in-human trial. And this is absolutely phenomenal pace of research. But the key and the really important thing to remember is that this has been preceded by years and years and years of research. So we want to take a step back and say, okay, how did we get to this point? How did all these decisions get made in such a short period of time? So very early on, of course, basically a lot of companies and a lot of decisions were made to focus on the spike protein. And why was that? Why did we pick the spike protein and not any of the other proteins in the virus? Well, the spike protein sits on the outside of the virus, and that's what allows this virus to attach to our cells and cause an infection. So this part of the spike protein on the coronavirus binds to a protein on our cells called ACE2. And so we know in respiratory viruses that bind to cells like this, we want to stop that from happening. If we can block this interaction, if we can prevent the virus from attaching to our cells, that's how we prevent an infection. And one of the best ways of doing that is producing antibodies. So as I mentioned earlier, those are the small proteins that can actually bind and attach to the virus and prevent it from sticking to our cells and prevent it from getting inside. So we knew from lots of research in influenza and SARS and other coronaviruses, this was likely going to be a good approach for making a vaccine against SARS-CoV-2. Now, that has its challenges though in and of itself. And I think this was actually a really fascinating part of the vaccine development that we didn't get to talk about very much. So the spike protein is something that we call a trimer, which means the protein itself is actually made up of 3 different parts that are identical and stick together. So this is a schematic sort of showing what we think the spike protein looks like. And each of the different colored sections represents one part of the trimer. So it's 3 different pieces that need to stick together and stay in the right shape. Now, the protein doesn't just stay in the shape though, unfortunately, because the whole role of this protein is to attach to our cells and allow the virus to get into the cell. So this protein actually wants to change its shape. And in these pictures here, you can see there are parts of this protein that move up and move down, And when the protein's actually done its job and allowed the virus to get into the cell, it's in a completely different shape. It's like a spring that's loaded up and then it wants to spring apart. And this can be really difficult for vaccine development when you have a protein that literally wants to come apart. The other thing is that this protein will actually get cut up into smaller pieces during the process of the virus infecting a cell. So this protein doesn't even stay together in one form during the entire process. So altogether, this is really tricky because we need to give our immune system an accurate and very specific target to raise an immune response to. But we have this protein that's in 3 pieces and it wants to fall apart and it wants to change shape and it doesn't want to stay together. So how do we actually design something in a vaccine that allows our immune system to see the way we want it to be seen? I'm going to stop sharing the screen just for a moment and show you my very sophisticated Lego model of the spike protein in an effort to sort of show you some of the challenges of vaccine development. So if we think about this, we have a protein that comes in 3 separate parts. So the first challenge is we need to hold those 3 parts together. In the virus, that's pretty straightforward and pretty easy because they sit in the membrane of the virus. But if you're going to make a vaccine, you need to figure out how are you going to hold these 3 parts together. Are you going to put it in a membrane? Are you going to put it in a virus-like particle? Or are you going to try to fuse these 3 pieces together so that they stay together in the proper shape? The next thing that we have to figure out is how we deal with all the moving parts of the protein. So this part that's most responsible on the spike protein for attaching to our cells can actually move around. And we say that it can adopt a down shape or an up shape. And so we have to find a way to stabilize that, and we have to know what we're showing to our immune system. The other thing, as I mentioned, is that these proteins want to fall apart. So it's very easy for each of these parts of the trimer to fall apart into different pieces. And we need to try to keep that together as much as possible. So scientists have developed methods where, very much like this Lego model, they can actually take block by block pieces of the virus and switch them out for other pieces. And then we can test them and say, okay, does this stay together better? Does this fall apart a little bit more? Can we make this really easily? And they can go block by block by block and replace each of those pieces. And there were groups at the NIH that had been doing this, particularly for the MERS coronavirus, or the Middle East Respiratory Syndrome virus, and other coronaviruses. And so they actually already knew exactly which pieces you should swap out to make the spike more stable. And it was that knowledge that allowed us to so quickly develop these versions of spike vaccines that were really, really effective. I'm just going to go back to the presentation here. So we needed to make a lot of decisions really quickly in terms of these vaccine development. We needed to decide what part of the spike protein we were going to use. And there were actually really early clinical trials that compared different parts of the spike protein. So we know a lot about the big RNA trials and the vaccine candidates that have gone forward, but there were actually many more candidates tested. Additionally, we had to decide how are we going to hold these pieces together. And that depends, and that tells you which different type of vaccine platform you actually want to use. And then finally, how will we stop the protein from falling apart? So most of the vaccines that went into development used some of those swapped out pieces that the NIH labs had actually shown to make a better, more stable version of the spike protein. And that was why they were able to move forward so quickly into their trials. Now, the other component of making a vaccine is sometimes we need to add an adjuvant. And an adjuvant is something that alerts your immune system and says, hey, you need to pay attention to this. And the reason that we need to do that is because vaccination and infection can actually be quite different. So in infection, there's lots of virus replicating throughout your body. You get very sick. There are lots of danger signals going on, and your immune system knows that something's wrong and it needs to make a strong immune response. However, when we give you a vaccine, sometimes it's just a small part of the virus. You don't feel that bad, and your immune system doesn't necessarily want to pay that much attention to it. So in that case, we add an adjuvant to just kind of wake the immune system up and say, hey guys, we need to pay attention to what's going on right now. In this chart, I just want to emphasize this was, again, partially why we were able to proceed so quickly with SARS-CoV-2 vaccine development. So there are a lot of different adjuvants that we can choose from. And as you can see, a lot of them have been tested in vaccines for a number of years. And all of these adjuvants in the COVID-19 vaccines that you can see here have been previously used in other vaccines. So for example, this one, which was used in one of the COVID vaccines, has previously been tested for influenza. There was another one that's been previously tested with hepatitis. So again, in most cases, we weren't using brand new components to make these vaccines. Instead, we were relying on the knowledge that we had from other vaccine candidates that would actually allow us to move forward quite quickly. Now, interestingly, the RNA vaccines actually get delivered in a nanoparticle that acts as its own adjuvant. So there are also exceptions to this. Not every vaccine actually needs an adjuvant. And again, it's through the studies of these different platforms that we've understood exactly how they react with the immune system. So where did that leave us? And again, I think we talk about the SARS-CoV-2 vaccines as though, you know, there's one vaccine or one or two vaccines that are the candidates. But in reality, a lot of different companies tested a lot of different platforms. And that just speaks to the fact that we didn't know exactly what was going to be best. So there were a lot of different approaches that were taken. So of course, there's been a lot of press attention towards the lipid nanoparticle vaccines of Moderna and Pfizer. But again, those vaccines haven't all been successful. There was actually a third vaccine tested called CureVac, which turned out to have much lower efficacy. And so we're learning from that about what makes a good or not quite as good RNA vaccine. Then we have the viral vector vaccines. So again, this is where we use a harmless virus to deliver information about the spike protein to our immune system. This is, of course, AstraZeneca, of the J&J Johnson & Johnson candidate, and also the Russian vaccine Sputnik. There were also vaccines that actually just used the inactivated SARS-CoV-2 virus. So this really classic vaccine approach that we've been using for hundreds of years. This was tested as well for COVID-19, and that includes Sinovac and Sinopharm. And then finally, we have protein vaccines. And these are the vaccines where we just pick that one particular part, in this case, the spike protein, and just deliver that to the immune system. And this was probably most well known as the Novavax vaccine, but it was also the approach that some of the Australian companies used, including the University of Queensland. So where do we go from here? We have a number of different vaccine candidates that have been approved, but what sort of lies ahead for the future of COVID vaccines? The first, of course, is the variants of concern. And again, this is a very well-known case right now, especially here in Australia and in Melbourne and New South Wales, where we have the Delta strain. So the question becomes, do we need special vaccines? Do we have to update our existing vaccines to try to combat these variants? And there's a lot of research going into answering this question right now, to understanding whether it's better that we just keep giving people boosters of the same vaccines that we have, or whether we should in fact be trying to develop new vaccines that will combat very specific variants. And that leads us into more questions about booster vaccines. How many boosters will we need? How often will we need them? Should we mix and match? When do we start giving vaccines to adults? And when do we start trying to make sure everyone has their first dose before we give second or third doses? And these are a lot of really complex questions right now, especially related to vaccine supply around the world. And finally, a topic that I think a lot of countries are still grappling with to some extent is the vaccination of young children, and how we can get more information from clinical trials, how quickly we need to look at vaccinating young children, and again, decisions around public health measures and the use of these vaccines just beyond the adult population. And then when we consider all of these things together, we still, I think, will have a lot of questions about how we live with this virus, how much we want to try to control it versus how much it will just continue to circulate around the world. And interestingly, there are a number of other coronaviruses that just cause the common cold and that do circulate worldwide. And we all get infected with these viruses in childhood. And I think it will be quite interesting to see what the future of SARS-CoV-2 is in that context. So just before we finish up, I just wanted to highlight a couple of things that I think are quite interesting about the vaccine development field moving forward. So one big thing in vaccines right now is this idea of nanoparticles. So really small, very sophisticated, formulated particles that can express different components of pathogens. And these have a lot of advantages. In some ways, they're much like the virus-like particle that I talked about in HPV, where they can express many, many, many copies of the same protein to really activate your immune system. But the other advantage is that we can mix and match different components of different strains on the same particle. So we could maybe put different parts of the influenza virus, but from multiple virus strains, all into one particle, all in one vaccine. And of course, there's a lot of interest right now in terms of whether we can do this for SARS-CoV-2 as well, not just with variants of the virus, but whether we can get a SARS and a MERS and a COVID-19 vaccine that takes care of all of those infections together. The other area that I'm interested to keep an eye on are alternate routes of administration. So right now we all get a jab in the arm, right into our muscle, one vaccine, quite straightforward. And there's a lot of benefits to giving vaccines this way, but there's also a lot of interest in understanding what would happen if we administered vaccines in other ways. So I mentioned some interest in the flu vaccine, where you can just sort of give it a nose spray right up the nose. And this is an interesting pathway to try to understand how we can get better immune responses at our nose, our mouth, and our other mucosal sites. Interestingly, there were also some pretty big landmark studies out in the last couple of years looking at what happens if we give vaccines intravenously. And this has been particularly of interest for TB infection, which of course, again, we're still looking for a much more effective vaccine. And there's also a lot of research around what happens if you give vaccines under the skin, and how giving vaccines in different ways can give you immune responses that occur in different places throughout your body and for different amounts of periods of time. So I think we have quite a while before we actually change how we give vaccines, but there's a lot of interesting research in trying to understand what the benefits might be of sort of changing up how we administer these things. And finally, I just wanted to highlight some of the really stubborn pathogens that we've been dealing with and trying to make vaccines for. And HIV, there are a number of challenges, of course, associated with an HIV vaccine. Part of the issue is that HIV actually attacks the immune cells that you need for a vaccine. And there's a huge amount of diversity across viruses that we need to deal with. But you might have seen in the news recently that there are RNA vaccine trials now going ahead for HIV. And in fact, a number of the COVID-19 vaccines were based off technology that had previously been tried for HIV. So there's been a lot of innovation and a lot of work done in science looking at HIV that's benefited a lot of other infectious diseases. Malaria is another big one, but there was actually a really nice success story recently in malaria. There was a vaccine that showed 77% efficacy in children in malaria endemic regions. And the thing that I thought was nice about this was that, again, this is that virus-like particle vaccine, but it used an adjuvant that's the same adjuvant that's used in the Novavax COVID-19 vaccine. So again, all of these products and all of these components are things that we're testing in multiple different ways. And just because one thing succeeds doesn't mean that it won't succeed also in a very different way in another vaccine. And then finally, tuberculosis, another infection that I've studied for some time, especially at the Public Health Agency. And tuberculosis can be really challenging to study. And we're still trying to learn a lot about the bacteria and which parts of the bacteria are important for immune responses. But like I mentioned, there's been some really interesting innovation, especially around giving vaccines intravenously for TB. And I think there's going to be a lot to learn in the next couple of years, hopefully spurred by innovation in the field. And so that's all I have for now. I'm very happy to take questions in the Q&A. And I just want to say thank you to everyone for joining in today.
Thank you so much, Jennifer. That was absolutely fascinating. The way you explain everything is just so clear, and I think I've learnt so much. And that Lego, I love it. I mean, your Lego skills, the slides, it was all fantastic. So thank you. And we've already got lots of great questions coming in, so I'll get started on that. And I guess everyone else out there, if you've got other questions, please feel free to add them to the Q&A function at the bottom. So I'll try and go through one very quick to start off with for Cooper. He was wondering, NIH, what does it stand for? And maybe we can add in what do they do as well.
Oh, sorry, yes, the NIH is the National Institutes of Health. It is the major government research body in the US. Sorry, that was my North American bias sneaking in there. So the NIH does a lot of really good vaccine research, and they're involved with Moderna particularly in developing that mRNA vaccine. And they have government scientists whose entire jobs are devoted to taking those Lego models and just replacing the bricks one brick at a time. And I feel very happy for those people, honestly, because it's not exactly the most glorious work reporting that you replaced 4 bricks and that worked better than 2 bricks. But it turned out to be a massively life-saving, world-changing piece of knowledge in the last year. So
absolutely. Well, if they've got models like yours, then we'll have a much better understanding about this amazing work they're doing. Great. So, okay, on to one from Howard. So he's asked, can you explain how the mRNA and DNA vaccines work?
Yes, absolutely. So the RNA vaccines, actually, I might just share my screen again. So the RNA vaccines have two main components to them. So inside is the actual RNA. So that is the genetic code, and that is the sequence for the spike protein. So that is basically a piece of instructions that we give to your cells, and the cell reads it and says, oh yeah, okay, I can make that. And then your cell goes ahead, it makes the spike protein, and then your immune system is like, wait, you didn't used to make that. Why are you making that now? What's new? What's up with that? And so that's how that works. And the important thing I think that people don't necessarily emphasize enough with the RNA vaccines is that those instructions don't last forever. They're very temporary. So once your cell has received those instructions, it kind of makes a bit of the spike protein and then it crumples the paper up and throws it away. So nothing goes into your DNA. Those instructions don't last forever. Your cells make the spike protein for a short period of time and then they kind of forget about it and move on. The other important part of the RNA vaccine is this shell that goes around it. And this is the lipid nanoparticle. So these are essentially small molecules of fats, kind of like cholesterol, really. And they form this little shell, and their job is to basically carry the RNA into your cell as safely as possible. So RNA is very fussy. That's why we talk about the cold chain storage for these vaccines. So they need to be kept very, very cold because the RNA is happy to just fall apart and degrade really, really easily. So this particle, this lipid nanoparticle, is a soft cushion basically that brings it into your cell, delivers the instructions, and then breaks apart and goes away. DNA vaccines are similar, although they're a little bit harder to deliver to your cells. And I think that's why the RNA products have gone ahead. They're much easier. You can just inject them with a regular injection. The DNA is a little bit more complicated. But yeah, the main points are the RNA does not stay in your cell. Nothing goes into your DNA. It's just a temporary instruction. Then it gets tossed and moves on.
Amazing, thank you. That was just really well explained. Yeah, and a nice soft cushion as well. Took that away as well. So thank you. Actually, I mean, even on that slide, I don't really need to bring the slide up, but there was CureVac as well, and you mentioned that was the mRNA type, but it wasn't as effective. So why do you know why that sort of didn't go ahead and why it wasn't as effective?
So we don't know for certain, but I think there's a fairly reasonably accepted idea. Basically, the RNA, so the genetic information in the Pfizer and the Moderna vaccines was modified in such a way that your immune system didn't attack it, I believe. So sort of like, we're going to read this, use the instructions, make a spike protein, but we're not going to try to destroy the RNA itself. The CureVac RNA was different and it wasn't modified in the same way. And that was probably the biggest difference between those two products. So right now we think that that might be why there was a difference in the effectiveness of the vaccine, but that needs to be tested. And so there'll be a lot of work looking now at, I think, different RNA modifications. And so again, that's sort of replacing different components and saying, okay, what works the best? But that's the best hint that we have right now. So that one, unfortunately, I think was only about 47% effective. It wasn't really enough to move forward with.
Yeah.
But I think that'll have pretty big implications for the RNA technology moving forward.
Yeah. I mean, I guess if it doesn't work, then you actually learn a lot more from it as well in terms of what will work.
It does in a way. It's kind of, yeah, I'm a bit skeptical of anything that seems like the magic bullet approach. And it's like, oh, RNA vaccines are going to be the cure for everything until the end of time. And that's would be lovely, but it's probably not just, it's not realistic. And so it's good when we can kind of see what really is the important key to making them as effective as possible.
Yeah, absolutely. Staying on the mRNA vaccines, now where was this one? There was someone that asked, oh, now I've lost it now. But basically they were asking about, recommendation or not in terms of combining them. So if we have one AZ, I say from Jimmy Giggle, but one AstraZeneca and one Pfizer, like yes, no, is that a bad idea and why?
It is, as far as we can tell, a good idea. And so from a scientific point of view, I was really excited to see when we started to get data out from those mix and match trials. And then also there's practical applications around, people not having to wait for the same type of vaccine as they received the first time. But all indications seem to be that it's either as good as getting 2 doses of the same vaccine or better. And especially in the case of AstraZeneca, because of the way it's delivered, because it's that virus vector, so we use that harmless virus to get the information into your cells. Eventually, if you see that enough times, your immune system can start to actually recognize that virus, and then it makes it harder for that vaccine to be effective. So there is potentially a benefit to sort of mixing things up a little bit and teaching your immune system things in different ways, getting it to pay attention again. And so far, that's been really successful. There might be slightly, so with the RNA vaccine, some people get decent side effects from them in terms of headache and feeling really tired and a bit of pain in the arm. That definitely still seems to happen when you have the mixing and matching approaches. But in terms of safety, it's a really similar safety profile.
Yeah, okay, right. And I just found out, so there was one person that was anonymous, so thank you, Anonymous, for putting that question in, but also Chris, and that was also from his son Orlando, who is 9. So it was a fantastic question, Orlando, thank you. I'll also just mention now, it is 2:45, so if anyone has anything to run off to, please feel free. The video of this event will be sent to you if you've registered for the event, and it'll also be available on the Faculty of Science YouTube channel as well if you want to catch up. But we've got so many questions, we might just go ahead with a couple more, Jennifer, because I know you're happy to stick around for a little bit longer. So we'll try and get through a few more, but everyone won't be going on for too much longer. Now, you started with side effects. Now, there was a question, I guess, about the extent of the side effects and why they change between different vaccines, and see if you can talk a bit about those side effects.
Yep. So it's been really interesting watching, as we have groups of people who have been vaccinated coming through our clinical studies and reporting their symptoms. So in general, I find people report more side effects with the second dose of the RNA vaccines. So it was quite common that people would say their first dose maybe felt like a flu shot, they had a sore arm, not much else. And then the second dose, they really sort of had that headache, tiredness, I'm going to go home from work a little bit early today. And conversely, I think it was the opposite with AstraZeneca. So people felt more side effects after the first dose. And I think it's related to how you're delivering information to your immune system. So again, the AstraZeneca vaccine relies on that harmless virus to sort of bring the genetic information into your cells. So in a way, there is kind of not an infection, but you are delivering parts of a virus and your immune system responds as such. And that's exactly what we want, but that does come along with side effects. The RNA vaccines, because we hadn't had a lot of large-scale human trials with the RNA vaccines, we were quite interested to see what the side effect profile was going to be. And I don't know that we have a full understanding of why the second dose seems to be more prone to side effects than the first dose. But I imagine we're going to see a lot more data on that coming out, but we still don't have a total understanding of what the difference is between those 2 doses.
Okay. Interesting. We've got another one from Anonymous. I believe it's a different Anonymous, but we just won't ever know. Interesting. So is there any way we can predict the evolution of the virus and therefore help to develop or update our vaccines accordingly? Good question, Anonymous.
That's a fantastic question. It's a question that scientists are also continually grappling with. So there are two things at least that are going on with the virus variants. One of the biggest things is that the virus, the mutations that we're seeing, allow it to bind much more tightly to that receptor on our cells. So I had that slide where it said, the virus attaches to the receptor on our cells, and that binding allows the virus to get in. So the mutations that we're seeing coming up are making it so that the virus can bind a lot more tightly, grab on more tightly to that host receptor. And so we can test for that, absolutely. We can say, okay, we take our little model, we change piece by piece, and every single time we can measure how well does the virus bind to that host receptor. So we can do that. The problem is those mutations also then have to be in a virus that is transmitted onto others, that creates enough virus in your nose that the transmission is ongoing, then it has to potentially evade your immune system a little bit and kind of be in the right set of circumstances to take over in the way that Delta has. And so we haven't become very proficient at the second part, unfortunately. So we can predict in the lab, yes, this mutation will make the virus bind more tightly, or yes, this mutation will make it so that our immune system can't see the virus as well. But getting that perfect sort of set of events all happening together, where things happen and there's transmission and it occurs enough that it becomes a global concern, is the part that we're really not good at. And so we've tried to do that actually with flu for many years. People have been trying to understand and predict in advance what the changes will be, and we just haven't been very successful. So people will keep trying, but unfortunately, we're just kind of missing that little piece of the real world transmission scenario.
Yeah, I mean, it's hard to predict the future, I guess, at the end of the day. You can find a lot of
So
options, but yeah, I know. Where's that crystal ball? I was going to ask, there's a few questions about, I guess, the boosters. There's a few questions came through, and I think you covered a lot of answers on that when you were going through towards the end. One from Leanne, do you think nasal sprays may replace any boosters that may be needed in the future for COVID in particular?
So I really like the idea of nasal sprays. Obviously, I think they're more user-friendly. Just the idea of not having any needles involved with things is always fantastic. It's obviously, really great for kids. We just haven't developed the technology yet to the point where I feel like it's become really, really, really effective. So we do absolutely want to generate immune responses right at the site of infection. That's absolutely the goal. But we haven't, again, quite figured out the formulation of how a nasal spray can do that. I think it may partially simply be because of anatomy. There's a lot of protective mechanisms in our nose from mucus and hairs and cells that basically try to stop things from getting in and try to not make our immune systems go absolutely crazy at that site. And so it's perhaps not the most effective way to actually generate immunity, even though we would think it would be. And so again, I think there will continue to be research into that space, but I don't feel like it's the next thing that's going to come out at this point in time.
Yeah. Okay. Well, it's good to have a bit of certainty at least. Now, there's a few questions here, and thank you, Leanne, for that question. Few questions here, I guess would come under, the worries around vaccines. A few here about the possibility of the vaccines, I guess, targeting our own cells and not sort of these foreign cells. I guess, what are the chances of that? And I guess, can you speak to that sort of worry that some people might have?
Yeah, so that's partially why I wanted to emphasize, I think, that these vaccines didn't come out of nowhere. So the viruses and the technology behind AstraZeneca has been developed and tested previously for HIV vaccines. These RNA vaccines have been tested previously. Novavax, with their adjuvant, has been making vaccines for flu and respiratory syncytial virus. So there's been actually a lot of data, and there was preclinical testing as well of vaccines for SARS back when the first SARS outbreak happened. And so we did have a sense already of how these things would work. We knew, again, that we wanted to make antibodies, that the spike was probably a good, something good for your immune system to focus on. And there's a lot of testing that goes into figuring out if pieces of these viruses are also going to elicit an immune response against parts of us. So there are techniques that we can do in the lab that basically check for things like that. And also, so there's monoclonal antibody therapy, which is like the Regeneron antibodies that President Trump received and things, where we give people sort of drugs for COVID. Those undergo the same testing. They make sure that they're not going to bind to things on our host cells. So there's a lot of things that go on sort of behind the scenes that are boring and nitty gritty and we don't talk about, but that all go into the testing of these things to make sure they're safe. And I think the other thing that I want to emphasize too is that sometimes the downside with clinical trials, even when we do a phase 3 clinical trial, is that it maybe enrolls 30,000 to 50,000 people. And that's a lot of people. It's a fantastic, it's a lot of money. That's a really good trial. And it gives us really a good indication of whether a vaccine is going to work or not. But if there's a side effect that only happens to 1 in every 400,000 people, it's probably not going to happen to anyone in a 50,000 person trial. And that's always been the case with vaccine rollouts. That's why we refer to the sort of early days of the rollout as phase 4, actually, because they're just things that you'll never know until it's out to millions of people. And that's why we have these systems in place for reporting of side effects and constantly sort of checking and going back and doing all these checks and balances. And so at the end of the day, we can do the very best that we can, but at some point it does become a numbers game, unfortunately, and those really rare side effects we're just not going to know until things roll out. And so I think it's important to be pragmatic and realistic when we communicate with the public about these things, and that we are really clear about what we mean about what is safe and not safe, but also still that balance that goes into the equation of deciding, how severe is a disease versus what is the benefit that we get from a vaccine.
Yeah, absolutely. So well said. Thank you. That's so clear. So yeah, so thank you for everyone that's sort of put in those questions. Got one here from anonymous number 3. Could the COVID and just I guess we're coming to the end probably, so we've got just a couple more questions, everyone. Could COVID vaccines provide any protection for other related coronaviruses, so SARS or the other SARS? Yes, like does that have a crossover effect? Are we double protected now?
It's a really cool question.
Yeah.
So we don't effectively, we don't know at the moment. I'm not aware of any studies actually, even in animals, that I can think of where we've given a COVID vaccine and then tried to give them SARS-1. We've done it the other way around. We've given them SARS-1 and then looked for COVID responses. So we do know that for our immune systems, if you're naturally infected, you can produce antibodies that recognize both viruses. And so people have actually, people that survive SARS-1 infection have become very popular in the last year or so. People have been looking at their immune responses, and you do make an immune response that can actually recognize multiple viruses. So that can happen naturally, but we also are very interested in making that happen on purpose. So there are some vaccine candidates now that are looking for pieces of all of the coronaviruses that are identical, and just kind of like that flu vaccine, just showing that one piece and saying, focus on this one identical thing, and then you'll recognize everything. Or vaccines like those nanoparticle vaccines, where we might actually combine pieces of SARS-1, MERS, and SARS-2 and try to give us sort of total immunity across all of them. So I think it's possible. I don't think we'll get equal immunity to all of the viruses at the same time. It's a bit of a challenge, but I would be very surprised if down the line we don't have what we would call a pan-coronavirus vaccine. I think that's definitely in our future.
Yeah. Okay, great. Look, there are so many questions here, but unfortunately we just don't have time to get to all of them. But thank you everyone for sending them in. I will finish off, Jennifer. I think we're okay to call you Jen now. I guess what's next on your, and you're saying when we first met to do this event, and then things are just happening so fast. And so, you know, what's on your to-do list for tomorrow is probably the best question, but I guess what's on your sights for the research? So yeah, what's up next?
Yeah, so we recently got a bit of funding actually to do some vaccine development ourselves. So we're trying to understand in that space around variants, basically, how can we get a booster vaccine that would give you the broadest amount of protection against many variants.
Yeah.
So we have that program going on. And then the other thing that I'm quite interested in is the data around people who recover from COVID and then get a vaccine, because the data suggests that those people have amazing immune responses, and they make these really high immune responses that are basically better than any other immune response that you can get from vaccines. And so I'm interested in understanding why that is, and what we can learn from that, and try to get everyone to have those amazing immune responses to vaccines in the future. So that's kind of where we're at in the next few months.
Yeah.
Days.
Yeah, exactly. Look, thank you so much for spending the time, for explaining everything. And sorry, everyone up there, we had so many questions and they were all really, really great. But hopefully we covered up on a few things and a few questions I guess you had. But look, it's just such an exciting area. And I think something you mentioned before as well, that there's almost been just as many papers published about COVID-19 as there are about HIV, which just blows my mind about the amount of work that's been done in this field. So I guess on behalf of everyone in the whole world, thank you so much for all the work, because we're very, very grateful, and hopefully we'll be out of lockdown soon thanks to some of your work. So that's about it. So again, apologies for going over. I know some of you would have had to drop out by now, but again, the video of this whole event will be up online. It'll be sent to you if you have registered for the event, but it'll also be on the Faculty of Science YouTube as well. And that goes for all the events that have happened over this week during Science Festival. There's a lot of really fascinating talks that happen. So I really encourage everyone to go over and have a look if you've got some spare time over the weekend, which perhaps if we're in lockdown, we might have a bit of extra time. So I think that's it from me. Thank you so much. I appreciate you tuning in. Have a great afternoon. And Jennifer, thank you so much. It was just, you're so well explaining, you're so good at explaining all these different really complex things. And it's been an absolutely fascinating talk. So, and your Lego skills, they're fantastic. Thank you so much. So have a great afternoon, and thank you everyone. Have a great afternoon as well.